As a comprehensive database related to rare disease research, the Rare Disease Data Center has developed AI-based genetic analysis tools, including Rna Splicer Prediction, mutation pathogenicity prediction, antisense nucleotide target prediction and more. The Rna Splicer Prediction tool may be used for predicting alternative splicing based on whole genome variation. Based on the deep neural network, this tool can effectively identify the mutations that cause aberrant splicing. Next, I will introduce how to use the Rna Splicer Prediction tool. First, you can type the key information of the gene. Taking the human DMD gene as an example, the gene information retrieved under this keyword is shown here. Some longer introns are omitted from the figure, and their coding regions are marked in green. The Rna Splicer Prediction tool provides three input options. G takes the gene sequence as the reference sequence, C takes the coding DNA sequence, CDS, as the reference sequence, and P takes the protein sequence as the reference sequence. I'll now show how to import mutations through different examples. If the user selects the mutation point through the cDNA location, the transcript ID needs to be selected. Users can fill in directly, such as C.65C mutated to G, or set corresponding parameters. When transcripts are used for reference sequences, there are various input methods. The mutation in the figure indicates that the 65th base of the CDS sequence is mutated to G. The mutation in the figure represents the 93rd position of the CDS sequence. And the base G of the intron sequence at the right 5 positions is mutated to A. The mutation in the figure indicates that between positions 52 and 53 of the CDS sequence, the original base AA is deleted and AGG is inserted. The mutation in the figure indicates that the base AGG is inserted between positions 52 and 53 of the CDS sequence. When the gene is used as a reference sequence, the mutation in the figure indicates that the 95th base of the DNA sequence is mutated to T. When the protein is used as the reference sequence, the mutation in the figure indicates that the number 3 aspartic acid in the protein sequence is mutated to histidine. The corresponding base GAT is mutated to CVC. As a result, point mutations can lead to several different splicing abnormalities, described as follows. Point mutations can lead to pseudo-exon insertions or exon skipping, generate pseudo-exons, and lead to partial deletions of exons in transcripts. In summary, the tool can be used to validate known mutation sites that cause pseudoexons, or to test whether the same mutation in the NCBI mutation database causes splicing abnormalities. Perform pathogenicity analysis of mutation sites in biomedical research, or predictive analysis of splicing abnormalities related to model construction.